today we are gonna, so now we've gone through just basic cell structure. Now we're gonna move on to how do these cells actually grow and how do they get the energy that they need to grow. So we're gonna go through a lot of terminology today in the beginning, but then we'll go through a little bit of mechanisms on how the cells get the different nutrients in the cells, and then we'll go through and defining the stages of growth and how you actually can actually calculate generation times and things like that. All right, so just in terms of general nutrition, right? So all the cells, they need certain things to actually be able to grow and to survive. So the essential nutrients, so these are the ones that they must have, but they can't actually make themselves, so they have to get it someplace else, right? So it's whether it's from the environment or they're getting it from another organism that they're living nearby, they have to be able to acquire these nutrients. The macronutrients, so these are the ones that they need in large quantities, so macro being large. So these are, the things that are in all of the major components of the cells, the lipid membranes, right? The amino acids, nucleic acids, those things. So those are the major elements that they need. They're also include these ions here. So these are the ones that, a lot of the ions, remember, they're like cofactors for different proteins and enzymes to function. So these are the major ones here. And then there's the micronutrients. So these are just trace elements, and they might be more specialized depending on the species of bacteria. But again, usually there's some kind of metal, right, that's necessary for some kind of enzyme to be able to function. But all of these things are required for these bacteria to grow. All right, so now we're going to learn some terminology. So essentially, all of the microbes that we're going to be talking about, they require electrons, energy, and the material to make cell parts, right? So carbon, that's what we will use as our marker for cell parts. And so we can actually, there's these naming structures to be able to categorize the different microbes based on how they get these three elements. So the first one we'll go through is carbon. So you can divide up how the cells get carbon into two groups, the heterotrophs and the autotrophs. So you can kind of think it as like top down versus bottom up. So the heterotrophs, they are breaking down organic compounds, so they're getting materials from somewhere else, and they're breaking all of that down until they get the carbon that they need. Versus the autotrophs, which is now going the other way from the bottom up, right? So now the simplest form of carbon that the cells can get is carbon dioxide. And so they're going to start with this carbon dioxide and then build up from that. And that's where they're going to get those carbon molecules. So some examples. So the E. coli, our favorite model organism, is a heterotroph. Autotrophs include things like algae and cyanobacteria. All right, the next thing is energy. And so energy to bacteria, we generally think of these as electrons. But you can categorize them in two different ways, so how they get the energy. So the phototrophs, they get them through light absorption. And then chemotrophs, they get them from chemical electron donors that are oxidized. So these we'll learn later in the class about these oxidation reduction reactions, and so that's how the chemotrophs are getting their energy. All right, and then the final one is electrons. So now essentially where the chemotrophs from the previous one, so where the chemotrophs are actually getting their electrons from. So the organotrophs, these are getting them from organic molecules, so things like sugars. And then the lithotrophs are getting them from the inorganic molecules, so things, these are inorganic chemicals, things like H2 gas, H2S, nitrate, things like that, okay? All right, so now it's in terms of naming. So we can just, you'll find in your book in a lot of, if you talk to just general microbiologists, they'll sometimes just, when depending on what topic you're learning, they'll just refer to these general names, and that'll just, it tells you a lot of information about the class of microbes that they're talking about. And so it always goes in this order. So the energy, electron, and then carbon, and then it ends in troph. Okay, so in these examples here, you have chemo-organo-heterotroph, chemo-litho-autotroph, photolitho-autotroph. And so if I were to give you one of these names, you should be able to tell me how each one of these are getting their carbon and their energy. Okay? There are some cases, like the chemo-organo-heterotroph, you'll often just see this as chemo-heterotroph or chemo-organotroph, because in this case, they're actually considered redundant, because if they're getting the things from the carbon, it's kind of obvious, right? So sometimes you'll just see 2 names instead of the 3 names, but that it's still you'll still get all of the information from that. All right, so now again, so regardless of source, the energy that they get, they have to store it somehow, right? Because you don't want to be in the case where you're just trying to get your energy on demand. You need to be able to have energy stored up, so if you need to make something right away, then you go ahead and you have this kind of energy storage to be able to do that. So the main ways that the microbes store energy is through chemical energy, so ATP. And again, we'll talk a lot about this later on in the course, but ATP is one of the major storage of energy, and this is, and something else that we'll talk about today, it's actually from this high energy phosphate bond. You might hear that term, and that's where the energy is coming from. The other thing is the membrane potential. So this, again, is generated when chemical energy is used to pump protons outside of the cell. So I've mentioned this term before, this proton motive force, which again will come more into play later on in the class. But you can actually use this chemical energy from ATP to generate a proton motive force using this ATP synthase here. So there is a pump that will literally take ATP molecules and use the energy from the ATP molecules to pump protons across the membrane. And that proton motive force can be used as a form of energy to do other things, right? So we'd already talked about the flagello, right? So that's one source. All right, so now we've kind of talked about, the different things that they actually need. So how do these microbes get the different nutrients? One of the more important ones to figure out is nitrogen. So remember, nitrogen is one of the macronutrients that they must have. So in our atmosphere, in the air is generally about 79% nitrogen gas, right? But essentially, most microbes can't actually use nitrogen in the gaseous form. It's useless to them. They can't do anything with it. It's actually very difficult to break down into gas. So they rely on other bacteria. So there are some bacteria, these nitrogen-fixing bacteria, that have the capability to use to convert nitrogen into ammonium with this special nitrogenase enzyme. And you see here, and we'll learn more about the cycle later on in the class, but essentially certain microbes have the ability to break down certain forms of nitrogen, and then other microbes have the ability to use these certain forms of nitrogen. And so they live in these communities and they can get the different parts from different places. All right? All right, so now how about the other nutrients? Okay, so you remember we just spent a bunch of time talking about the cell membrane and how it is this semi-permeable barrier, but if you look at it, a lot of the things that they need, it can't actually make it past through the membrane, right? So they need ways to get these things. These are things that are essential to them for survival. But if you also remember the other component of the cell membrane that we talked about, there's a lot of proteins, right? And so some of these proteins are these permeases and pores. A lot of them are substrate specific. Some of them, their specificity is pretty broad and can allow in a lot of different kinds of molecules. But in general, this is how a lot of those other nutrients are actually making their way into the cell. Okay, so the first one that we're gonna learn about is facilitated diffusion. So diffusion is just, you know, molecules moving in past the membrane, but it's always moving down its concentration gradient, right? And so in facilitated diffusion, it's moving down the concentration gradient, and this actually doesn't require any energy. So these molecules, so in this case, in this picture, it's glycerol, right? Glycerol is one of those things that can't freely pass through the membrane, so it requires this protein to do it. So there's specific proteins in the membrane that allow glycerol to come through. So glycerol is going to come in and it's going to bind this protein here. Once this protein actually sees and it's actually bound up the right substrate, so glycerol in this case, it's going to cause this conformational change here in the protein. So the proteins will change and fold in different ways depending on what's like actually interacting with the different amino acids, right? And so that conformational change then is going to essentially let it spit the glycerol back out the other side of the membrane. Yeah.
When we say down their concentration gradient, that means it's different than their own concentration.
Just meaning, so from high to low, so what's on the outside versus what's on the inside. So it's going to go from high, if it's high concentration on the outside, then it's going to go in because it's low on the inside. So if it was high on the inside and low on the outside, you couldn't use this mechanism. All right, so the other way of nutrient uptake using these different proteins is active transport. So the facilitated diffusion, again, it's moving down the concentration gradient, so, and it has these specialized proteins to help it do it, but it doesn't require any energy. In this case, now that's pretty rare. A lot of these things will require energy for it to come, for it to pass the things into the cell. So the first case are these symports and antiports. So this is coupled transport of molecules. So it's gonna, so in this case, you have two different molecules that are gonna be moving, okay? One of them is gonna provide the energy for the other one to come through. So the one that is moving essentially down the concentration gradient is gonna provide energy for the one that needs to move across up the concentration gradient. And so in general, so you might think, so, well, why, if, you know, there's already lots of it in the cell, why would it still trying to get more? The cell is actively going through growth, metabolism. It needs a lot of these molecules. So even if there is some in the cell, it just might be in an environment where there's not much on the outside, but it still needs it, right, to be able to do what it's doing. So there are cases where even if they have a lot in the cell, it's still going to want the things to, so it needs ways for it to move against the concentration gradient. Okay, so again, so in the symport, now they're moving both in the same direction. Okay, so you have the two molecules coming in, and they're both coming out on the same side. Although the one from the red one in this case is providing the energy for the blue one to cross through. Okay, and then as the name suggests, the other one is the antiport. So now they're going to be moving in the opposite direction. So in this case, you have the red molecule coming in. And then, so it's going to cause that conformational change. And then as the red one is coming out, it's now in this state where it can accept the blue one. So the blue one will come in, and that will go and spit out on the inside. And so now you've transported the two molecules, but now in the opposite direction. All right, another form of active transport, so again, requiring energy, it's using another type of membrane protein system setup, and this is called the ATP binding cassette superfamily, so ABC transporters. So these are important both for uptake, so things coming in, so it'll bring in things like sugars and amino acids, and then they also use these pumps for efflux, so things like drugs, so if they're common cause of antibiotic resistance, right? So if you are trying to treat this bacterial culture with these antibiotics, the antibiotics are gonna come in, they're gonna be like, this is bad news, I don't want this, and so it's gonna try and spit it out. If it spits it out, then it's not gonna be able to take effect. Also, same thing for hazardous chemicals or for any kind of byproducts that it's making in metabolism that might be harmful to it. It'll have a way to spit these things out. So the thing about the ABC transporters, as the name kind of suggests, it's using ATP. So again, the high energy phosphate bond, so this phosphate bond here is providing energy for it to move a molecule across the membrane. It's very common for these to have, in this picture, as you see, this solute binding protein. So it has these extra proteins within the system to help kind of lock up and find the molecule that you're trying to move. And so it will do that, and it kind of helps associate with the protein in the membrane just to make that transport easier. All right, so the last class of active transport that we're going to talk about today is this group translocation. So this one, it uses energy, and it actually alters the substrate as it's coming into the cell. So what it was on the outside and what it is on the inside is going to be different, because it's going to actually chemically alter that on its way in. And so technically, it's moving down its concentration gradient, but this still requires energy to do so. So the major class in bacteria for this is the phosphotransferase system. Okay, so this system uses energy, so not from ATP, but from phosphoenolpyruvate. So this is also known as PEP. Same idea though, it has this high energy phosphate bond. Okay, so it's using the energy from PEP to actually bring in molecules. In this case, it's usually bringing in some kind of sugar molecules like glucose, mannitol. Okay, so how this works. So inside the cell, you have PEP. Okay, not in this picture, but there are other pictures. There are multiple pictures describing the system in your book, but you'll see that there are a couple of other components that it actually uses to take this phosphate bond and pass it to several different proteins. So this phosphate here is getting transferred from this PEP molecule, and eventually it ends up here on this enzyme 2B here that you see. Okay, so what happens now? The cell needs glucose. So glucose is going to come in, and it sees this protein here, this 2C, is specific for glucose. So it's going to let glucose only in. And then on the way in, glucose is going to get modified with this phosphate. So now it's going to become glucose 6-phosphate. Okay, so what was on the outside is no longer what it is on the inside. So it's technically moving down its concentration gradient. But again, it is still requiring energy from one of these phosphate bonds. Okay. Now there's actually about 20 different versions of this for different carbohydrate molecules. So these are kind of what we call modular, and so while the 2B and 2C proteins for these are going to be different for every single sugar that's coming in, the molecules that are in here that are not in this picture, that are getting the phosphate bond from PEP, those are the same, and so those can be used across all 20 different ones. But you'll have a specific protein in the membrane that is going to recognize the different carbohydrates out here. Any questions about this? Yeah.
Did the phosphate fall off that PEP, or is that whole P that whole structure?
Yeah, so there are going to actually be, I think it's two different, so there are actually like two different steps here. So the phosphate gets passed from this to this protein here. So there are enzymes that are actually going to come and rip the phosphate off, then transfer it to another thing, and transfer it, finally transfer it to this. Any other questions about this? There's also, yeah.
Oh, actually, what's the most common like method that usually the body, or there is no most common one?
You mean just for transport in general?
For like transport.
So every type of thing is going to be different. So it's very specific to the molecule that needs to come in. There are some cases where they're kind of like generic things that kind of let in a lot of different things, but like in these cases, they all have a specific binding pocket that can bind different kinds of molecules. So it's very specific. I also posted a video of this system on Canvas, which is pretty useful. All right, so as you notice, we skipped section 4.3, so the methods for culturing and counting bacteria. We're covering all of that information in the lab. But the things that we want to note from here that moving forward, so the kind of concepts that we're talking about for growth conditions and things, I'm all going to be talking about referring to like laboratory conditions and not necessarily environmental conditions. But so one kind of aside as far as just, again, more into like historical and microbiology and where we're at. So, you know, after about 120 years of trying to grow these microbes in the lab, we only know how to grow about, 1% of them, right? So there are lots of different microbes out there that we are just not able to grow in the lab. And there are a lot of people that make careers about just trying to figure out how to grow one microbe. Because they all have very interesting and important value to us, but it's hard for us to study them if we can't grow them ourselves. But so how do we know that these actually exist? So again, it goes back to the ribosomal RNA, right? We know that every microbe has this ribosomal RNA, and thanks to the the invention of sequencing and PCR, so we find a lot of these different microbes basically just by taking environmental samples and doing 16S sequencing on them, and we get all these different sequences, and then we know the ones that exist in our lab that we can grow, and then you compare them and you're like, this is something new. And then there are all these phylogenetic trees, which we'll talk about towards the end of the class. But so now people have these databases and databases of this information of these microbes that we know that they exist, but sometimes we can only study them in their natural context, which is a little bit more difficult. Yep.
So you take the soil sample or the water sample and then you sequence it and then all of them, and I guess you get rRNAs for all of them?
Yeah, so you can actually, in reality, in what you're doing the DNA, but so you're PCRing off the DNA and the genomes, but you're getting information about the ribosomal RNA itself. Yep.
So you get the rRNA, but then you're like, something's there, but I just can't see it because it doesn't grow in the petri dish.
Yeah, so you know that you get all these different sequences, right? You know that they're not the same, so they're representing different species of microbes, right? And then you can compare them to databases of existing ones, but you know that like you can only end up growing a certain number of them, and then you see all these sequences that aren't actually growing, and then so that's just how we know that they exist, but we might not know how to grow them. And then people can spend time trying to figure out they're like, oh, but this sequence is super interesting, so like I have to figure out how to grow it, and so they'll take time to try and do that. All right, so now we're going to talk about the growth cycle. Okay, so most bacteria, they divide by what's called binary fission. Oops. All right, so it's going from, you basically get one cell that's going to dividing. So we talked about cell division last time. So you have one cell that's going to divide, and you're going to end up with two cells. And then those two, they're going to go and they're going to divide again. You get two more cells each. Okay, so this process is called binary fission. And then each time something divides into two, that's considered a generation. So this is considered a generation, and then this is considered a generation. Okay, so what we want to try and figure out is ways to calculate how many cells we have in a culture that we're growing. And so if n represents the number of cells, so the total number of cells, so like the total number of cells in this population here is going to be proportional to 2 to the n, the little n, where this little n is essentially number of generations. Okay, so in this first step here, right, so we had 2 to the 1, so there's 1 generation, right? So we ended up with 2 cells coming out here. And then if we were going to count both generations, we have 2 to the 2, which is now 4 cells, right? So we have 4 cells here. Okay, so now, but what if we had started with 10 cells, right? We didn't start with 1, we started with 10. So now we have to adjust the equation, and so we get n of t equals n0 times 2 to the n. Okay, so this nt here, this is the final cell count. The n0 here is the initial number of cells. And then the little n is still the generation time, right? So now again, so if we were started with 10 cells, so we would have 10 cells, which would be the n0. That's how much we started with. Okay, and then times 2 to the n, let's just still go with doing 2 generations, right? So now we would end up with 40 cells instead if we had started with 10 instead of 1. So this equation here is key, and so this is how we calculate the number of cells. And now, so this is only for exponential growth, and we're going to go through different growth phases in a minute, but so just so you know that this is for exponential growth, this equation. Any questions about this so far? Okay, so the next thing now that is useful is to actually be able to figure out how many generations has happened. Okay, so if we have this equation, now we actually want to just solve for the number of generations. So that's this n, the little n. So how we're going to do this when we have an exponential in there, so we're going to use logs. So if you take the log base 2 of both sides, and then remembering your logarithmic rules. So again, so if this is multiplication, when you take the log, it becomes addition. And then the exponents now just look like this, right? Okay, so this was basic, just taking the log of both sides of the equation. Okay, so now this log base 2 of 2 essentially is 1. And so now you just get this is equal to, and now we're going to move everything onto the other side and solve for n. All right, but now again, remembering your rules of logarithms, this subtraction here can be combined into it becomes division. And so now we have, right, so this is the number of generations. So if for some reason you knew how many cells you started with and how many cells you ended with, you should be able to calculate how many times did the bacteria divide. So how many generations were there? Any questions about this? Sorry, what?
On the exam, you're going to use like the exact equation, or we have to get there?
I don't. You're gonna have to get there. Yeah. Sorry, what?
So we'll need to know how to solve that on the exam?
Yeah. So I'm not gonna give you the equation on the exam.
Will we need like a calculator for the loans?
Nope. So in reality, I will have you solve these things conceptually. You won't actually have to plug in numbers. Yes.
So are we saying that we need to learn how to derive it, or
This is just for your information, so you know how this is derived, right? So if you forgot what this equation was, and all you remembered is the first equation, which is the most important one, then you know how to get there, right?
Okay.
All right, so this is for number of generations, right? But what actually we kind of care about more is the actual generation time. So I've been telling you things like, oh, E. coli doubles in about 30 minutes, right? So that's what the generation time is, right? So what we really care about now is solving for generation time, so which is the doubling time. Okay, so the equation for generation time, so that's G is equal to T over N. So T is essentially the growth time. So how long you are letting your culture grow, okay? And then N is the same thing from before, the little n, so a number of generations. So this has units essentially of time, right? So because it's time per generation at the end. Generation is unitless, and so this has generation, so it's generation time, right? So it has units of time. Okay, so this is going to vary with species. Okay, so it's going to vary with what they're growing in, so the media, the conditions, the temperature, pH. And then you just remember that this is also for growth in pure culture, so only with one bacteria in there, right? Because if you have multiple species in there, they're going to interact with each other, and that's going to be different. So these equations only apply for pure culture, right? And then kind of the last terminology that you kind of hear with respect to bacterial growth is the growth rate. So the growth rate, I think your book uses K to represent growth rate. So growth rate, so it's just related, it's proportional to the inverse of generation time. And then, so the units of this, so the units of K is essentially 1 over time, usually represented as like inverse hours or something. All right, so we are gonna I'm gonna have you think through a problem and try it out. So look at this problem. So if you put 10 to the 6 bacterial cells in broth, how many cells will there be after 2.5 hours of growth if the generation time is 30 minutes? So we're gonna pretend that it's all exponential growth. So I want you to think about how you're going to solve it, talk to your neighbors, figure out and explain to them how you would actually solve this. I'm not going to be looking for numbers. I'll go through it. I'll walk through it after, but I want you to think about it first. So given the equations that we just went through, how would you solve this problem? So you decide, as long as you're consistent, right?
Okay.
So as long as you're consistent. So. Mm-hmm. All right, so I'm going to walk through this with you, okay? So hopefully you've all thought about this yourself. Okay, so how do we go about to solve this? So what are the equations that we have, right? So we have these two equations. So we have the equation for the total number of cells, and then we have the equation for generation time, right? Okay, so what have we been given in the problem? So we have to figure out what these numbers are telling us, right?
Growth time and generation.
And generation number of cells.
Yep.
We have T, the total amount of time that is allowed, and we have N, which is the amount of our reproduction time. But you have to convert your minutes and your hours into the same units.
All right, so we don't actually have N, we have G. So this is in hours, and then we have G, right? So this is 30 minutes. Okay? So now we can use this information to solve for N. So as long as you were consistent, so I'm going to convert to hours. This is 5, right? And so now we have all the information we need to solve for this. All right, and so that's all I would ever expect you to do on an exam. I don't care that you can plug things into a calculator. So that would be the final answer.
What would be the growth rate?
So the growth rate, essentially is the inverse of this, but in reality it's only proportional to that, and it depends on. So you'll learn this in the lab section later on in the course on how to actually calculate the growth rate.
So we won't be asked to calculate it. Okay.
Nope. All right. Okay, so now we're going to move on and talk about the different stages of growth, and the key thing in this is that this is for batch culture, okay? So batch culture is a closed system. So these are things that you're doing in lab. So recently in lab you did aseptic transfer and you inoculated the different broths, right, with the Serratia cultures, right? So that tube with broth in it, when you grow that, that is considered a batch culture, okay? It's closed system, what goes in, and then that's it. So it's it's key to know that those equations that we were just using is for batch culture, and it's only for this exponential or log phase. So that's where those equations are representing. But so these batch cultures, they're very good and easy ways to study physiology and growth of bacteria. You can change different things in the media and see how they affect it. You can add add and subtract different components one at a time, and so it's a very nice and easy system to just understand how the different microbes grow. Okay, so as far as the individual different phases, so there are 4 different phases. So the lag phase. So the lag phase, the cells are not yet dividing. Okay, so again, this is kind of a condition that we're creating, right? So we're making this up. So the same thing that you did in your aseptic transfer, you took so from a slant, so you had cells on like a hard medium, right? That you then transferred them into this liquid broth. So now this broth is a very different media, right? So the cells were probably they're actually in stationary phase by the time you got them, and you transfer them. Now they're in this brand new place. There's all of this nutrients available. It's in a different kind of media, different kind of conditions. So now the cells are in there, and they're essentially adjusting to that. Okay, so they were in stationary, now they're turning on different genes, turning off different genes, getting ready and adjusting to the nutrients that exist in the broth that you put it in. So another way that you could do is you could also test what happens to bacteria when it goes from this kind of broth to this other kind of broth with different nutrients in it. And so again, when you transfer them, they're going to go into this lag phase, and they're going to start and adjust to the new conditions that they're in. Okay, so then the next phase here, so they're not actually dividing here, they're just getting ready to divide. The next phase here is this log or exponential phase. So this is where they're doubling at that kind of constant generation time that we just calculated. They're not synchronized though, so you can have one bacteria that's slightly off from another one, and so you are getting things doubling at different times, but they're still kind of doubling at this rate. So this is how we get this exponential curve here. Okay, and then towards the end, so kind of in this transition region towards like late log phase, as it starts to hit to stationary, the cells are essentially, the rate that they're growing at slows. And then when they hit stationary, so they have stopped growing for one reason or another, whether it be that they've run out of nutrients, right? Because of the batch culture, you only have certain amount of nutrients in the media that you provided them. Or it could be that it's making some kind of toxic byproduct from the metabolism that it's doing, and that's actually causing growth to slow. And then eventually, right, if that continues for a long enough time, the cells are actually going to start dying because there's just nothing left for it, and they're going to start dying. So they actually die again in this kind of half-life with a negative exponential. So again, on this log plot here, it looks like a straight line here because it's actually dying at an exponential rate as well. Any questions about this? All right, so that was batch culture, okay? So that's in contrast to continuous culture. So continuous culture is a way for us to essentially trap cells in logarithmic growth. And so they reach this, what we call a steady state. So you can have this extended period of logarithmic growth, and how you actually do that is by having ways to essentially remove and add new media to your culture. Okay, so and then you have to adjust at actually how the rates of the new media that you're adding and the media, and so you're going to remove media plus some of the cells too, right? So you're going to have to adjust the rate that you're adding new things and how much you're pulling out, and it's going to all be based on the generation time, so that because you don't want it to get to a case where you're taking out more bacteria than you're making, and because then it's just going to mess up your growth. But there are systems that you can use to create these kinds of conditions. They're very useful, again, for studying different kinds of physiology. You can also study kind of evolution experiments in there. You can just put them in there for an extended period of time and see if you can try to trigger some kind of evolution if you're kind of trying to kind of strain the bacteria under certain conditions. And so I guess what I was describing essentially is what we call a chemostat. And so your gut is essentially a chemostat, or essentially because you're bringing in different foods and going through your intestines, and then it's coming out as waste, right? So it's like a real life chemostat, as long as you're eating properly. I am going to stop there because we won't have time to finish the next topic.